So let's take a look at some of the specific experiments that Mendel conducted that led him to this understanding, to this idea that there are dominant alleles or dominant traits and there are recessive traits. Okay, I mentioned already one experiment that he did where he took true breeding tall plants and he crossbred them with true breeding short plants. In this case, he was following a single trait. In this case, the trait he was following was height of plants. And he did several experiments like this where he followed a single trait. He did plant height, he did color of the flower, he did shape of the seed, he did color of the seed, etc., etc. Every time he did an experiment where he followed a single trait, this was called a monohybrid cross. He would cross a purebred, true breeding version of a tall plant with a true breeding version of a short plant, for example, a monohybrid cross. And then what he did was he carefully counted up how many tall and short plants he had in the first generation, and then he crossed members of the first generation together and then counted up in their offspring how many tall and short plants that he had. And what he found was, again, in the first generation, the F1 generation, 100% of his plants were tall, and that in the second generation he had 75% of his plants being tall and 25% of his plants being short. And here's why. In the parent generation, the P generation, we had a true breeding tall individual, TT, and we had a true breeding short individual, little t, little t. These are diploid genotypes, aren't they? Because they contain an allele that that plant received from its mother and an allele that that plant received from its father. When these plants make an egg and sperm cell, those egg and sperm cells, remember, are haploid. So each egg and sperm cell can contain only one allele. In this case, there's only one option. This plant can only make gametes with a capital T in them. This plant can only make alleles with a small t in them. So if I cross a egg with a capital T in it with a sperm with a small t in it, the union of these two together will create a zygote that is a heterozygote. And indeed, that's what we see here in the first generation. All of the individuals in the generation were heterozygotes and they were all tall. Now if I take two of these heterozygotes and cross them together, which is exactly what Mendel did, something interesting happens. Again, these are diploid genotypes. This individual, when it undergoes meiosis, can make a gamete with a capital T in it, or it can make a gamete with a small t in it. But there can only be one or the other in each gamete if it's going to be a haploid cell. Same over here. I can either have a big T in the cell, the sperm or egg cell, or I can have a small t in the sperm or egg cell. So now I got to figure out, well, what happens if this egg cell unites with that sperm cell, or what happens if this egg cell unites with that sperm cell? Well, the easy way to do that is to draw something called a Punnett square. And in a Punnett square, I'm going to put one parent along the top. Usually the female is put across the top of the square. Her gametes are put across the top, and the males are put down the side. Or you can reverse them. Some purists say that there's only one way to do a Punnett square. I personally don't care. If you want to have the father's gametes appear across the top of the Punnett square and the mother's gametes appear along the side, I don't care. As long as one parent's along one side, the other parent's along the other, I'm happy. Then you're going to fill this in. I'm going to take the information from up here and just fill it in down the column. Then I'm going to take the information from here and fill in across. And in this case, I'm going to do this a little differently here. It's just customary to usually write the dominant allele first if it's present. It just makes it easier to read the genotypes. So you can see here when I do this cross, I get one individual that's homozygous dominant. I get two individuals that are heterozygous. Here's my two heterozygotes. And I get one individual that is a homozygous recessive. And what that means is these three individuals all will have a genotype a phenotype that appears tall. This individual will appear short. We get a 3-to-1 ratio of tall to short. He found this time and time and time again when he did monohybrid crosses. Okay. This second slide here just shows what I did on the last slide. I had made myself room here on this slide and ended up didn't use it, but this is where we showed we get this 3-to-1 ratio. So let's go ahead and move along here and talk about how Mendel interpreted this ratio. What he said was, basically, anytime I get this 3-to-1 ratio in the second generation offspring, that must mean that I have traits that are dominant and other traits that are recessive. And the way they are interacting with each other is determining whether I get tall or short offspring, or whether I get offspring with purple flowers or white flowers, or whether the pea pods are round or wrinkled, or whatever it is. We now know what Mendel called traits are actually what we call alleles now, different versions of genes. It's pretty amazing to think that he came up with all of this stuff before we ever even knew what DNA was at all. He was able to work this out. Now, once Mendel had this down, he started to make things a little more complex. He wanted to know, well, what happens if I follow two traits simultaneously? What's going to happen? This is what, when he followed two different traits to see how they sorted out in the offspring, this was called a dihybrid cross. Last thing we learned about was a monohybrid cross. Now we're going to look at a dihybrid cross. So what he did was he looked at, he took, for example, a true breeding tall plant that made green pea pods, and he bred it with a true breeding short plant that made yellow pods. And he wanted to see what distribution, tall and short, green and yellow, he would get. And he found out that the traits, as I said before, sorted independently of each other, and that just meant that they were located on different chromosomes. Now, I want to take you through step by step. Do I want to do that here? Let's do it on this one real quick, and I'll show you how we did this. So we had a true breeding tall green plant here. That means that its genotype was big T, big T, big G, big G. This one was a true breeding short yellow plant, little t, little t, little g, little g. This individual can make gametes. Every gamete has to have at least one allele that relates to plant height, so it has to have one of these two, and every allele has to have at least every gamete has to have at least one allele that relates to the color of the pod. So we have to have one of the T's and one of the G's in each of the haploid gametes. And you can see the only combination we can get is a big T with a big G. There's no other possible combinations for a gamete from this plant. Versus over here, the only combination I can get is a little t and a little g. And when I combine those together, I get all the F1 generation having a big T and a little t, and a big G and a little g. Make sure you write the T's together and the G's together like that. It just makes it a lot easier to be able to see what you're dealing with. And this is a true heterozygote. It's a heterozygote for both height and for seed, for pod color. And this individual, all these individuals in this first generation were tall, and they were green in terms of the kind of pea pods they made. What happened when we cross them in the second generation? Well, I'm going to let you see if you can't figure that one out for yourself. We are going to work through some of these more complex Punnett squares like this in just a little bit. So if you get stumped on this when you try to work it out on your own, don't worry, we will go over it together so it clarifies it in your head for you.